No, it's not a bad problem, but I don't want to go with them until I get everything worked out, so that when I hand them back to you, everything's good and I don't have to take them back. Okay, let's change the title of this. Should be Lecture 9. Okay. Save. All right, there we go. So today we're going to do cell physiology. We finished up with cell biology, the structure of the cell. Now we are concerned with the function of the cell. And this is where, unfortunately, the chemistry lectures will come back to haunt you for the first of many times. Okay, plasma membrane. Going all the way back to the first lecture, this is going to be what separates the inside of the cell from the outside of the cell, and having boundaries is one of the characteristics of a complex animal. Plasma membranes regulate the passage of substances into and out of the cell. Okay, we can take things in, we can put things out. Plasma membrane is able to detect chemical signals from other cells that allows it to communicate with its environment, as well as the plasma membrane can act as an anchor to adjacent cells. So we know that cells come together to form tissues, and one of the ways that happens is when cells start to interact with each other through their plasma membranes. Here are a couple different pictures of the plasma membrane, better known as the phospholipid bilayer. So we talked about phospholipids in the chemistry lecture under the lipid heading. And bi means two layers of phospholipids. That's what your plasma membrane of most of the cells in your body. So over here on the left, you have the real picture, scanning electron micrograph there, the two dark lines indicating the phospholipid bilayer. In the middle, here's the artist's rendering. Okay, we see the phospholipids and we see many large, colorful proteins lodged inside the phospholipid bilayer. And then over here on the right is just the chemical structure. Another picture trying to give you a more three-dimensional aspect to the phospholipid bilayer. Again, here's one layer of phospholipids with the head and two fatty acid chain tails, phosphoglycerol head, two fatty acid chain tails. That makes up the phospholipid bilayer. Okay, phospholipids. Hey, sorry. It's all right. Yes, I do. All right, hold on. That's all right. Okay, back to phospholipids. So phospholipids again, phosphoglycerol head, two fatty acid chain tails. That's a single phospholipid. It has a polar region. What's another word for polar? Hydrophilic, meaning what's its relationship with water? It likes water. So the head is the polar region. It can interact with water. Phospholipids also have a nonpolar region. What's another word for nonpolar? Hydrophobic. And those are the fatty acid chain tails. Because of this orientation, that one end of the phospholipid likes water, the other end does not like water, they will organize themselves so that the fatty acid chain tails are together and the polar heads are together. The parts that like water hang out with each other. The parts that don't like water hang out with each other. Okay? Remember that you are mostly made of what? Water. So mostly the outside of your cells is water, and mostly the inside of your cells is water. So we would expect that the heads are going to be in contact with the water. So we would see heads facing the outside of the cell and the other layer of heads facing the inside of the cell. This is the only thing that holds the phospholipids in, what's the word I'm looking for? In contact with each other. Okay, there are no chemical bonds, there are no ionic bonds, there are no covalent bonds. It's just the fact that one part of these phospholipids likes water and the other part does not like water. So if I just draw my ball and stick model here, and this is the outside of the cell, and this is the inside of the cell. The heads line up so that They like water. Inside of the cell is mostly water. Second layer, heads line up because the outside of the cell is water, and they can handle water. So in this situation, the farthest that you could ever get away from water is right here. And that's where all the fatty acid chain tails that don't like water hang out. Okay, so this is what leads to the orientation of the phospholipid bilayer. Another way we describe the phospholipid bilayer is by calling it the fluid mosaic model. Okay? Fluidity is an ease of movement. Okay? If something moves with great fluid, that means it moves very easily. What's a mosaic? What's a tile mosaic? It's a picture made from lots of little pieces. Okay? If you're looking at a tile mosaic, you'll see a bigger picture, but it's made up of a bunch of small tiles. It's a mosaic because it's one big membrane, but it's made up of individual phospholipids. And then model, again, it's just a model we have, the fluid mosaic model. And what that refers to is the ability of this membrane. It can bend, it can wiggle, it can change shape, all without doing damage to the cell or disrupting any of the structural integrity of the cell. So again, no bonds here, no covalent, no ionic bonds, nothing like that holding those together, just the association of hydrophobic and hydrophilic, and that's really what allows it to bend, to wiggle back and forth, to let pieces go, to take pieces in. It's very forgiving in its shape and structure. Okay. More specifically, we talked on the first slide about some of the functions of the cell membrane. Here are some more specific ones. Border or barrier, again, separating the inside of the cell from the outside of the cell. It's a receptor site. Okay? Receptors allow cells to communicate with their external environment. They're just proteins. They can sense hormones outside the cell. They can sense neurotransmitters outside the cell. Autocrine and paracrine factors. What does auto mean? Self. Auto means self. An autocrine factor is one that the cell makes. And although it doesn't make a bunch of sense, sometimes cells will secrete a factor and then it will come back and act on the cell itself. Para means beside. So other cells, other tissues in the area may have released a factor. That would be a paracrine factor that can be detected by the plasma membrane. An antigen site. Some of the proteins lodged in the plasma membrane allow your immune system to tell the difference between a cell that belongs and a cell that doesn't belong. Okay, self or non-self. Every cell in your body, well, almost every cell in your body, has a protein on the cell surface that says, I belong as part of this body. If a bacterial cell, if a virus comes into your body, they do not have that protein. And that's one of the things that helps your body to identify them as foreign. And finally, transport. Okay? Ions, water, nutrients, oxygen, carbon dioxide, all of these things have to pass in and out of the cell. And in order to do that, they must interact with the plasma membrane in some way. Okay, membrane proteins. We keep saying proteins lodge in the membranes, proteins are receptors, proteins are this, proteins are that. So membrane proteins will break down into two categories, what are called integral membrane proteins and peripheral membrane proteins. If something is integrated, what is it? If two things are integrated, are they? They're together, okay? And peripheral, where is your peripheral vision? It's around the side, okay? Just without knowing anything else about what an integral peripheral membrane protein is, we already know something about them, okay? Integral membrane proteins are closely associated with the membrane lipids, with the phospholipids. The phospholipid bilayer is integrated with the integral membrane proteins. They are closely associated. Most integral membrane proteins will span the phospholipid bilayer. That means they'll have one part on the outside of the cell going all the way through another part on the inside of the cell. What holds them in association are the polar regions of the protein associate with the polar regions of the plasma membrane, and the nonpolar regions of the protein associate with the nonpolar regions of the phospholipid bilayer. So here's my integral membrane protein. It's spanning the phospholipid bilayer. It's just wedging itself in here between phospholipids. What can you say about this area of the protein in terms of polarity? It's either polar or nonpolar right here. It's polar because this part of the protein is going to associate with the polar regions of the heads. So what can we say about this area of the protein? It's nonpolar, it's hydrophobic. And then this area of the protein, hydrophilic. Remember when we talked about proteins, we said that they become so large that parts of them begin to act independently of one another and that they can fold and change their shape according to hydrophobic and hydrophilic interactions. This is the case where we have a protein that's so large it has several different regions. It has hydrophobic regions, hydrophilic regions, and when it integrates into a phospholipid bilayer, hydrophilic regions stay with hydrophilic regions, hydrophobic regions stay with other hydrophobic regions. What this does, again, there are no bonds, there are no covalent bonds, there are no ionic bonds, is it gives the ability for this protein to move laterally. If you move laterally, what direction are you moving? You're moving side to side. This protein can move side to side because that maintains its association with polar and nonpolar. But what if all of a sudden this protein tries to move up or down? What does that do? If I shift this protein up, that's going to bring this polar region where? Into a nonpolar region, and that's not going to work. Okay? Same thing for shifting it down. Proteins cannot shift up and down because they're bound by that polar-nonpolar interaction. But they can move as much as they want side to side because that keeps still the polar and nonpolar regions in line with each other. Some proteins won't move at all, and that's only because they are anchored by another protein located inside the cell. So not all proteins have the ability to move laterally. Some of them will be anchored. And here's a picture. So the three that we see here right in the middle, the purple, the green, and the purple, these are all integral membrane proteins. They are spanning the phospholipid bilayer. Okay, and even the small ones here to the right and to the left are integral membrane proteins. Even though they don't span the complete membrane, they are still closely associated with it. Where do you see peripheral membrane proteins? There are two on this picture. Peripheral means on the outside, on the side. Any guesses? This line on the inside, this light blue line and this darker purple line, those are peripheral membrane proteins. They are only appearing on one side of the membrane. Even if they were to appear on the outside of the membrane, what polarity statement can you make about peripheral membrane proteins? They are completely polar or hydrophilic. This protein has to be able to totally interact with water because it's on the inside of the cell and it's mostly water, and it's also forming an association with the already polar heads of the phospholipid bilayer. So while integral membrane proteins have both polar and nonpolar regions, peripheral membrane proteins will be only polar. Yeah, it could be on the inside or the outside. So transmembrane proteins are a class of integral protein. Trans means what? If I go trans-America, what am I doing? Going across America. So a transmembrane protein is one that crosses the membrane usually several times, okay? 5, 7, 9 times these proteins will go back and forth. They serve several functions. Usually transmembrane proteins are responsible for transporting ions or water in and out of the cell. They are able to transmit chemical signals from one side of the cell to the other, so from inside to out or outside to in. And they are so large that even though they could move, they usually do not, so other proteins may anchor to a transmembrane protein. So here's an example. If we start here, we see that it crosses 1, 2, 3, 4, 5, 6, 7, okay, and then it has what's called a cytoplasmic tail coming down into the inside of the cell. So this protein right here is lining up in the same fashion as any other integral membrane protein with polar and nonpolar interactions, but it is so large it will probably not move. And so other proteins could choose to anchor to this protein. Peripheral membrane proteins, again, do not associate with the nonpolar region of the phospholipid bilayer. We already said that peripheral membrane proteins are completely polar. They are only hydrophilic. They love water, so they will stay away from the nonpolar fatty acid chain tails. Because of that, you will usually find them on one surface or the other, extracellular or intracellular. Extracellular is, what's your best guess? Outside the cell. Intracellular is inside the cell, also called cytosolic. So when you see the word intracellular or cytosolic, that's something inside the cell. Extracellular means outside of the cell. A lot of times peripheral membrane proteins will associate with other membrane-bound proteins. They could associate with those transmembrane-bound proteins or other cytoskeletal elements. There is a structural element to the inside of the cell, and peripheral membrane proteins may associate with those and help influence things like the shape of the cell or the movement of that cell. And again, here are a picture. We have this one. The two that they are illustrating here are both intracellular or cytosolic proteins on the inside of the cell, this light blue to the right or this purple to the left. Those are both peripheral membrane proteins. Okay, we talked a little bit before about diffusion. Why was diffusion so important when we talked about it in cell biology? It was the reason for something. Anybody remember? Diffusion is the reason that cells are so small. Diffusion is the reason that cells are so small. Okay? And we said we would talk more about diffusion, so here it is. What is diffusion? Every particle, which is a solute, and it does not matter if it's in gas, if it's in water, it doesn't even matter if it's in a solid. Every solute wants to be evenly distributed. Okay? So without a barrier or any type of regulation, okay, we're not manipulating anything, we're just letting particles and solute go where they want. Solute will go from places of high concentration to low concentration until it's evenly distributed. A wonderful example of this is the simple air freshener. Okay, if I were to grab an air freshener and bring it in and plug it into the wall right here, okay, at first it would smell good right here. But then I could back away and eventually the whole room would smell like the air freshener. The air freshener is a high concentration of that scent molecule. But since I'm not regulating it, since I bring it into the room and I let it spread out, it's going to go from an area of high concentration to an area of low concentration until it is evenly distributed, till the entire room smells like air freshener. That is the same for anything, okay? Even the desk particles, okay, the particles that are making up your desk. That's a high concentration of desk particles, where your desk is. But if we leave it there for long enough, it's going to disintegrate because those particles want to go from an area of high concentration to low concentration. Same with the glass, same with particles in water. If we dump sugar into water and we don't stir it, eventually it will dissolve because the sugar particles want to go from an area of high concentration, which is in the crystals, to an area of low concentration, which is into the water. All solute will behave this way. This process is known as diffusion. Almost every living organism depends on diffusion to move molecules important for life. You depend on it for carbon dioxide, for oxygen. Okay, that's what gets those substances in and out of your cells. Just diffusion, just the fact that oxygen wants to move from an area of high concentration to low concentration, and so does carbon dioxide. There are three types of diffusion: simple diffusion, facilitated diffusion, and what's called active transport. Simple diffusion is what we refer to when we just say diffusion. Okay? Very important characteristic, it does not require any ATP. What is ATP? It is energy. What does it stand for? Adenosine triphosphate. And what macromolecule group does it fall into? Nucleic acids. Okay? Getting you guys ready for the exam next week. So under simple diffusion, okay, no ATP, solutes will simply move down their concentration gradient from an area of high concentration to an area of low concentration. It can occur in a region or through a membrane as long as our solute can pass through that membrane. So what I'm saying is, if I hung up a big sheet in the middle of this room and I put the air freshener on this side, okay, the scent can move through the sheet. So that's a membrane, but our scent molecules can move through the sheet, so it can move through a membrane as long as that membrane is permeable or allows that particular molecule to pass through. Again, oxygen leaving your bloodstream and going into the cells, oxygen concentration in your arteries is high, so oxygen wants to move out of the arteries and into the cells where it's low. Carbon dioxide builds up in the cells because of the mitochondria. It's low in the blood, so carbon dioxide wants to move out. This all happens through simple diffusion. Facilitated diffusion also does not require any ATP. So right now we don't have any way of telling the difference. Solutes are still moving down their concentration gradient from high to low. Still does not help us to tell the difference between the two. Here is the difference, this third bullet point. Facilitated diffusion has to employ something. It has to use something. Usually a channel, a gate, a transporter. Something has to open up for that solute to come through. Simple diffusion does not have that requirement. Facilitated diffusion does. An example is glucose. Glucose has to move into your cells. And what is glucose? Before we go to sugar, good. Glucose is just sugar. Sugar has to move into your cells through a glucose transporter. It can't just move through the membrane. So when your blood glucose is high, the glucose channels open, they allow sugar to come into your cells. All right? Does not require any ATP. The opening and the closing of these channels does not require any ATP. It just simply moves from high to low through a channel, through a gate, through a transporter. The last one is active transport. Number one difference, this does use ATP. Okay? That's the biggest difference between active transport and the other two. It uses ATP because it's transporting a substance uphill against its concentration gradient. It is taking solute from where it is low and it is trying to put it into an area of high concentration. The only way that can happen is by a constant supply of ATP. This is like trying to roll a boulder up an endless hill, a hill that goes on forever. If you ever stop holding or pushing that boulder, it's going to roll right back down the hill. And that's exactly what active transport is. Your body must expend energy to push molecules in a direction that they do not want to go. They do not want to go into an area of high concentration. So if that energy ever fails, if we ever stop, all the molecules come flowing back out. Okay? ATP use against the gradient, so when we use ATP in an instance of active transport, it's usually coupled with an ATP producing movement. Okay? An example is what's called a sodium-potassium pump. Okay? One molecule is moving down its concentration gradient, and you can think of it's spinning a wheel. And every time it spins that wheel, it produces an ATP to help bring a molecule back in. Let me draw a picture. One molecule gets to move down its concentration gradient, and it's coupled to a mechanism. You can think of it like a spinning wheel, and every time this wheel turns, it makes one ATP so that we can bring another molecule back into the cell. Okay? And this is a very common mechanism when we have to use active transport. We will couple it with the movement of another molecule that's not using active transport. Here's a artist rendering of the three different types of diffusion. Here's simple diffusion in the top panel. The black represents a membrane, but it is permeable to our molecule. And we know in simple diffusion, our molecule will move from an area of high concentration to an area of low concentration. Same movement in facilitated diffusion in the middle, except this green dot is representing some sort of channel or gate or transporter. It's facilitating the movement from high concentration to low concentration. And finally, the bottom panel, active transport, notice the arrow is going the opposite direction. We are going from low concentration to high concentration. And this one uses what molecule? ATP. Okay, so we talked about the movement of a molecule. Now we're going to talk about the movement of water. Osmo is water. An osmoreceptor can sense water. Osmosis, movement of water. Osmolarity, concentration of water. So osmo is always going to be water. The net diffusion of water across a membrane is the definition of osmosis. Osmosis is facilitated by aquaporins. Why can't water just move right through the plasma membrane? Because there's a large part of the phospholipid bilayer that is hydrophobic. Remember those inner fatty acid chain tails. There's no way they're going to let water come in there. So we have to have some sort of pore that allows us to bypass those fatty acid chain tails. These three words are incredibly important. They're going to help you through any osmolarity question I could ask you. And when you get into 202, you can completely understand the function of your kidneys if you can just remember those three words. Okay? Water follows solute. Wherever the solute concentration is highest, whether it's in your body or in a glass of water, that's where the water will go. It will move into that area in an attempt to equilibrate the solute. Okay? So wherever the solute is high, that's where the water goes. Okay? Water follows solute. While we do not think of water as having a concentration, it does. I can have a glass of 100% water. If I start to add stuff to it, if I add sugar or salt or a tea bag, I am lowering the concentration of water. Okay, so you have to remember that water itself also has a concentration. Okay, we're going to step away from water for a minute. I want to talk about other transport mechanisms, and then we're going to come back to osmolarity at the end. Other transport mechanisms include endocytosis and exocytosis, and we already talked about what those words mean. If endo means to what? To bring in. Cyto is cell, osis is movement. Endocytosis. Endocytosis is to bring something into the cell. Exo means to leave or to push out of the cell. Cyto, osis, okay, movement of something out of the cell. So endocytosis is when a region of the plasma membrane folds in on itself and pinches off to produce an intracellular membrane-bound vesicle. Similar to the way that we saw when we talked about the two prokaryotes swallowing each other. Remember that when the small prokaryote came in, it was wrapped in a piece of plasma membrane. That's a type of endocytosis. Something comes into the cell, it pushes on the plasma membrane, and then the plasma membrane will pinch off and produce a little vesicle. Exocytosis is the complete opposite. It's already bound in a piece of membrane, and it joins up to the existing plasma membrane and releases its contents outside the cell. So here's a picture. These little blue dots are coming into the cell. So the gray line here is the plasma membrane. It pushes in, and then it will pinch off and produce a little vesicle. And here's the opposite. These little purple dots want to leave the cell, so they are packaged in a piece of plasma membrane. They join up with the existing plasma membrane and are expelled to the surface. Now here's the question. These little purple dots that are already bound in plasma membrane, where have they just come from? They wanted to leave the cell. So they had to rendezvous with one of the cellular organelles in order to get packaged correctly. There, the Golgi. These purple dots must have just come from the Golgi, because that's the only way to leave the cell. Visit the Golgi, get packaged in a piece of plasma membrane so that you may exocytose yourself out of the cell. Endocytosis, bringing something into the cell, can come in two forms: pinocytosis and phagocytosis. Pino is a liquid. Okay, so when pinocytosis occurs, it is the bringing in of a liquid or other type of adsorptive material into the cell. Phag or phage, and you'll hear it both, okay, means to eat. Okay, you have phagocytes in your body. Those are cells that will eat up other cells. Okay, when endocytosis occurs with a very large particle, like a bacteria or some type of cellular debris, that's phagocytosis. Okay. Exocytosis is important in its own right because it serves two main functions. It provides a way to replace portions of the plasma membrane that were removed due to endocytosis. Okay? And if we get to add new membrane components, it's like we get to constantly refresh that plasma membrane. We get to add new parts to it. Okay? It also provides a route by which membrane impermeable molecules, okay, molecules that cannot naturally move through the membrane by themselves. And what category would the molecule fall into if it can't get through the membrane? Polar, nonpolar, hydrophobic, hydrophilic? What type of molecule cannot get through? Polar. Anything that likes water can't get through because, oh, I erased it, because it can't deal with the fatty acid chain tails. So any polar or hydrophilic molecule, a hormone or something like that, that the cell makes can only be secreted into the fluid, into the extracellular fluid, if it goes to the Golgi, becomes packaged by the Golgi in a piece of plasma membrane, and then is exocytosed. What can you say, now that we know that endocytosis and exocytosis are opposites? One takes a piece of the plasma membrane, one gives a piece back. What can we say about the rate of endo and exocytosis? They have to be the same. They have to be happening at the same rate. What if more endocytosis happens than exocytosis? What's going to happen to the size of our cell? Endocytosis. If more endocytosis is happening, I keep taking away pieces of the plasma membrane during endocytosis. So what's going to happen to the plasma membrane? It's going to get smaller. If I keep taking away pieces of the plasma membrane, it's going to get smaller. If I exocytosed all the time, if I kept adding pieces of plasma membrane, then what would happen to the cell? Now it would get bigger, in terms of just speaking about plasma membrane. Okay, variables that can affect membrane transport. And again, just like variables that can affect chemical reactions, we're talking about speed here. How fast or how slow can we move a molecule in or out of a cell? It's going to depend on 4 things. The first of which is going to be solubility, whether or not that molecule is polar or nonpolar. Polar or water soluble, also known as hydrophilic substances, such as water, will diffuse very slowly or not at all, because they're going to run into what? They're going to run into the fatty acid chain tails in the phospholipid bilayer, which will not allow them to move. Nonpolar substances, on the other hand, lipid-soluble or hydrophobic substances, diffuse very rapidly. Okay? Nonpolar molecules just push the phosphoglycerol heads aside. They dissolve in the fatty acid chain tails, move through, and get into or out of the cell. So again, polar or nonpolar can determine how fast or slow that cell or that molecule can move in and out of the cell. Size, large versus small. Okay? Assuming all other properties are the same, we're dealing with 2 hydrophobic molecules or 2 hydrophilic molecules. Okay? Large molecules will move slower in relation to small molecules because large molecules have more surface area. And more surface area means more resistance, and more resistance makes it harder to move. Compare a muscle-bound man, okay, a weightlifter, he's 300 pounds, he's the most muscular man in the world. He's very, very strong, but if you put him in a pool full of Jell-O across against a tiny little wiry, thin 100-pound guy, the 100-pound guy is going to have a lot easier time moving through the Jell-O than the 300-pound guy, even though he's stronger, because there's less surface area, there's less resistance, it's easier for him to move through the Jell-O. And you can think of your cell as Jell-O because you're mostly gelatinous on the inside of your cells. It's cytoplasm. Pore size, how big or small the pore is. So we know that only polar ions or molecules are going to need pores. So if they like water, if they are hydrophilic, they need to have a pore. How big is that pore? Can that pore let 10 molecules through at a time, or can it only let 1 molecule through at a time? That's going to determine how fast or slow that substance moves. And last one, molecular charge. Okay? Again, it applies to ions, which are charged. Some ion channels may have walls that will attract or repel ions based on charge. So if I am a positive ion, I carry a positive charge, and I want to move into the cell, what will the charge of my pore be? It will be negative because opposites attract. Okay? So if I'm a positively charged ion, I will move faster through a negatively charged pore than I would a positively charged pore. Okay? And this second bullet point here is just planting a seed for when we get into neurons. The side concentration gradient, once we start dealing with charges, we introduce something called membrane potential. And membrane potential is separating electrical charges across the plasma membrane, putting all your positive charges on one side and all your negative charges on the other. That plasma membrane is blocking them from being attracted to each other. Okay? Ions of the same charge will repel each other, so positive will repel positive, and negative will repel negative. Okay? And again, I'm just planting a seed here because that's going to become important when we talk about neurons. Okay, back to water and osmotic pressure, and this is the last thing we're going to talk about today. So the pressure exerted against the membrane due to the diffusion of water is called osmotic pressure. Okay? But unfortunately for us, we can't measure osmotic pressure. So we have to measure something else called hydrostatic pressure. So here's my beaker, and it's filled with water. And I'm going to put a membrane in it, okay? And this membrane is permeable to water. What does that mean? Water can move back and forth across the membrane freely. And I'm going to put a high amount of solute here and a low amount of solute here. Which way is water going to move? To the right compartment or to the left compartment? But water does what? What's our saying? Water follows solute. Wherever the solute is the highest, that's where water will go. So water is going to move through this membrane into the compartment to the left. As water is moving through this membrane, it's exerting a force against it. That is osmotic pressure. But we can't measure that directly. So what we do is measure the amount of force we need to stop it. Hydrostatic. If something is static, like stasis, there's no change. Okay? So once we can stop the water, hydrostatic pressure, so we exert a pressure that we want to stop it from moving, that is hydrostatic pressure. Okay. So we measure osmotic by determining the hydrostatic pressure needed to stop osmosis. Therefore, osmotic pressure only represents, it's not a direct measurement, it only represents, the amount of pressure that would have to be applied to the solution to prevent the passage of water from one side of the membrane to the other. Why is this even important? Why are we talking about this? Biology of osmotic pressure. All of your cells have a membrane, so water will move in and out of your cells according to solute concentration. Cell volume can increase and decrease according to the concentration of solute. Okay? And we have different words to describe different solute concentrations. So I'm going to draw myself 3 containers. Okay, each one is filled with a solution. This solution will be what's called hypertonic. This one will be called isotonic, and this one will be called hypotonic. And inside of each one of these beakers, I'm going to plop in one of your red blood cells. That's not red. Okay. Red blood cell, red blood cell, red blood cell. And I'm going to tell you, and this is on the very next slide, that the solute concentration inside of your red blood cell is about 0.3 osmoles. So let me just put a 0.3 inside of each one of your red blood cells. Okay. Hyper. What does hyper mean? Fast or active? High. It means higher. Okay. In this instance, it means higher. So a hypertonic solution has a very high solute concentration. So let's make it 0.7. Okay. ISO. Anybody know ISO? equal, or the same. So if it's got to be the same, I'm going to make it 0.3. Hypo means what? Low. Okay? It's got to be low. Let's make it 0.1. So I have given you the solute concentration inside of the cell and the solute concentration in each one of the containers. Now the question becomes, what is water going to do? Let's start with hypo. Where is the water going to go? Into the cell. There's going to be water moving from the beaker into the cell. As a result, what's going to happen to the cell? It's going to start to swell, and eventually it is going to burst, or what we call lyse. Okay? The cell will lyse. It will explode. Isotonic. What's going to happen under those conditions? No change. Okay? Really what happens is the same amount that moves into the cell is equal to the amount moving out of the cell. Okay? And finally, hypertonic. Which way is the water going to move? It's going to move out of the cell. There's water inside the cell and water outside the cell. So water is going to move outside the cell, and that is going to cause that cell to shrink, or what's called crenate. Okay? So the osmolarity of your blood versus the osmolarity of your blood cells is incredibly important. Let's say we're out on the football field and one of the football players passes out because he's dehydrated. Well, if you're dehydrated, what are you missing? Water. Well, I'm just going to take a big old jug of water and I'm just going to stick it right into his veins. And I'm just going to start pumping water into his blood. What's that going to do to him? Why is it going to kill him? No, it's not a question of oxygen. If I start putting a bunch of water into your blood, I'm diluting your blood. I am bringing you into a hypotonic state where the concentration of the solute in your blood becomes low because I'm diluting it, and the concentration inside your blood cells is high. I would explode all of the red blood cells in somebody if I simply put water into their blood. Even though they're dehydrated and they're missing water, you cannot put water into somebody's blood because this will happen. Whenever you get an IV bag, okay, it's always a sugar or a salt solution with an osmolarity of about 0.3 that matches your red blood cells, so that if somebody puts liquid directly into your blood, it's under isotonic conditions. Okay? That's why osmotic pressure is important. You always have to remember what's going on in the blood versus what's going on in the blood cell. And again, water follows solute. If you know where the solute concentration is the highest, you could figure out any possible scenario I threw at you. Okay? If I tell you two solute concentrations and ask you where's the water going, if you can remember that water follows solute, you can figure out where the water will go. Okay, and again, the osmolarity of an average human in their red blood cell is between 0.27 and 0.3 osmoles. Any questions about this lecture? Okay.